Hello, my name is Lily Karbowski, and this is my exam mini video number 4. Watson and Crick were scientists in the 50s who discovered that the structure of DNA included base pairing between nucleotides, which allowed for constant width to be upheld in the helix shape. The hydrophobic nucleotide bases are in the center, while the hydrophilic backbone is on the outside. There are 5 hydrophobic bases: adenine, thymine, guanine, cytosine, and uracil. Adenine and guanine are purines with a 2-ring structure. Thymine, cytosine, and uracil are pyrimidines with a single ring structure. Each purine pairs with a pyrimidine, which allows that constant helical width to be maintained. Adenine pairs with thymine via 2 weak hydrogen bonds, and guanine pairs with cytosine via 3 weak hydrogen bonds. Uracil is only present in RNA and pairs with adenine in place of thymine. The main stabilizing force for the structure of DNA are the hydrophobic interactions. The hydrophilic phosphate backbone is water-loving and is happy to be in contact with the water inside our cells. However, the hydrophobic bases are water-hating and avoid water at all costs. Think of the DNA as a spiral staircase. The hydrophilic phosphate backbone would be the handrail that shields and surrounds the steps, and the hydrophobic bases would be the steps protected by the railing. The way in which nucleotide bases stack on top of each other maximizes the hydrophobic interactions that keep DNA together. Each base has 4 ribose carbons. The 1' carbon attaches to the base. The 2' carbon distinguishes DNA from RNA. DNA has just a hydrogen attached, while RNA has an OH attached. The 5' carbon has a phosphate group attached, and the 3' carbon is what bonds to the 5' carbon of the next base.
DNA features major and minor grooves that alternate throughout the helix. The major grooves allow more space for the DNA strand to interact with proteins. Watson and Crick's DNA is right-handed, meaning that the strand coils up and to the right. DNA strands are antiparallel, with one strand running the 3' to 5' direction and the other running the 5' to 3' direction. If you were to put DNA into a higher salt concentration buffer, you would increase the DNA's midpoint of thermal denaturation. This is due to the salt shielding the negative charges on the phosphate group of the DNA's backbone, stabilizing it further. When the negative charges are not shielded in a low salt environment, the electrostatic repulsion makes it energetically more favorable for the strands to pull apart.